And I'm glad to kick off this new series. I hope it will be very successful. In picking a topic, I felt that I wanted to talk about something I've been working on for about a year and a half now, but on and off, of course. But I put in a few opening slides about protein structure, not being quite sure who exactly will hear my presentation. So let me start with a few thoughts about why we're so excited about folding, right? And the main reason is that proteins are essential components of the DNA protein world of today through their chemical activity. They essentially do all biological catalysis. Nucleic acids are the repositories of information, but their only role in life nowadays is to determine which proteins get made when, where, and in what amounts. Everything else is done by proteins. So in order to do catalysis, proteins need to assume a three-dimensional structure, and that is what we call the folded structure. And while nucleic acids fold pretty robustly, and you can denature them and renature them many times over, as you know from the PCR reaction, with proteins, it doesn't work, right? If you boil an egg, then you get a hard-boiled egg, and then when you cool it down, you have a cold hard-boiled egg. You don't get the proteins back which aggregated in the egg. And so proteins from the beginning were seen as a much more difficult object of study than nucleic acids. And as we gradually realized through many trials and errors, the free energy of folding of most proteins is just a few kilocalories per mole, which is typically the energy of a good hydrogen bond. So a folded protein has an energetic advantage over an unstructured protein of just a few hydrogen bonds. And that's what makes it so excruciatingly difficult to deduce structure. And people have banged their heads against this problem for decades. And in fact, I also participate in CASP contests, and I didn't see any progress for almost 20 years. And so I concluded that this problem will not be solved in my lifetime. And yet now we not only have AlphaFold, but we have language models, and we're making huge strides in accessing protein structure in the way we accessed protein sequence starting, in the 80s. Now, why is protein structure still a major problem, even given that now we have so many tools to deduce it? The answer is that most random chains of amino acids do not have a folded structure. So this is quite different from nucleic acids, where you can pretty robustly expect the nucleic acids to assume some sort of structure. Proteins typically don't. And the estimate I wrote here of 1 in 10 to the 10th is the absolute upper boundary, which is given to us by large-scale experiments, as have been done by Andreas Plückton and Jack Szostak and Greg Winter. And they typically didn't get more than one success in libraries of 10 to the 9th to 10 to the 12th exemplars. And so clearly, random chains of amino acids practically do not fold. So it's a huge mystery how nature could come to be built on folded proteins and use them for all its catalytic needs. And here in Tübingen, when I set up the department, one of the most important questions that I had was, if domains are the unit of protein folding and are too complex to have arisen just by chance, how did we get them? How did we get the proteome that is substantially composed of domains? Now, I knew that most protein domain families go back to prototypes that were present in the last universal common ancestor. And so it showed that the creation of new folded forms had by and large ceased. But that in biology is not necessarily surprising. If you look at body plans in living, multicellular living beings, we don't really have new body plans coming up. The last big episode of body plan creation was in the Cambrian, 500 million years ago. And so with proteins, it looks like we're also not creating new protein families. And the question is this due to the fact that it's difficult to get folded proteins, or that the folded proteins we get are not useful and they cannot survive against heavily entrenched, established protein domains? And I'll touch on this at the end of my talk. And for now, I want to take you to how we think domains arose, and then how the power of repetition seems to have shaped the proteome more strongly than almost any other phenomenon. So let's move ahead. My hypothesis here in Tübingen was that the folded proteins arose from peptides that had been optimized in the context of RNA-based catalysis and replication, the so-called RNA world, and that these peptides acquired the ability to form structure in the context of a scaffold. Now, why the RNA almost quantitatively went away, we don't know, but I suspect a major catastrophe which wiped out the RNA, for example, by raising the temperature on the surface of the Earth. But whichever way it was, at some point, peptides had to start doing the catalysis that had ancestrally been done by ribozymes. And the most useful peptides in this evolutionary step were the ones that were already cofactors of ribozymes. And so we thought that in principle, we might be able to find these ancient peptides by comparing modern day proteins in sequence and structure. And here is the sequence alignment, which then gave us the structures that we wanted to analyze further. And we use structure only to validate similarities that we found in sequence space. And in this way, we felt more confident that we were tracking homology. And in the process of doing this, we developed a vocabulary of peptides that is by no means exhaustive. And it's certainly also not all the peptides that played a role at the origin of proteins. It's just the ones where the signal of homology is still strong enough that we could convince ourselves and reviewers that we were on the right track. Now, there's a lot of things to say about this, but that would be a completely different presentation. The one point I want to address here are the fragments encased by dotted lines, right? Because all of these form their fold by repetition. And we were struck by how pervasive repetition was in our ancient peptide vocabulary. But we shouldn't have been surprised. If you have spent any time with the history of protein science, you will know of the paper by Dayhoff and Eck, Margaret Dayhoff being one of the true giants of early protein bioinformatics. And Dayhoff and Eck proposed that ferredoxin evolved by consecutive duplications of a tetrapeptide. Now, I don't think they are right, but certainly ferredoxin in the last step evolved by duplication of a motif, and that was the most significant signal they had in their paper. But in 1966, with, forget some 5 or 6 protein structures known, their ability to point to repetition as one of the dominant factors in the origin of folded proteins is stunning. It's amazing. So if you haven't read their paper, do take a look at it. It was in Science in 1966, I believe, Dayhoff and Eck, a truly amazing step. And fairly soon after them, Andrew McLaughlin at MRC started to outline the power of repetition in generating protein domains. And he spent some 20 years and as many papers describing one event of duplication or amplification after another. And so, as I say, I shouldn't have been surprised that repetition is so powerful. In fact, experimentally, almost all the laboratory investigations of the formation of protein structure worked by repetition in TPR repeats, in beta propellers, in beta barrels of the outer membrane. There are, and of course, in coiled coils. Coiled coils have been engineered since the 80s, following the pioneering work of Bill DeGrado. And they are, of course, the quintessential repetitive protein domain. So anyway, repetition is everywhere. And so when we set up an expectation of the methods that, or the mechanisms that we would see giving rise to folded proteins, one of the truly first and prominent ones was repetition. In contrast, recombination here, for example, the recombination of these 2 fragments to give you the structure over there, recombination has not worked for us or for anyone else. We have only been able to recombine these 2 fragments by making 18 point mutations in the sequence. So we mutated about a quarter of the total sequence in order to get a folded exemplar. No one can tell me that evolution does such things. You need to get fairly quickly to a selectable form or you can't get on at all. And so recombination has not worked for us. And every time I go to talk to colleagues in this field, they all say, oh, it hasn't worked for me either. Right, but repetition we used in the cases I named TPR repeats. That's the crystal structure of a TPR repeat we generated from a ribosomal hairpin. We generated beta propellers, we generated membrane-embedded beta barrels. These are all things that worked in a straightforward way with at most a small number of mutations needed to stabilize the fold sufficiently for experimental determination. And so repetition, of course, is rampant in genomic DNA. Almost everywhere you look, there's scores and scores of repetitive regions in DNA, most of which are considered not to be expressed or not to be coding. However, prokaryotes are also full of repetitive DNA, and in prokaryotes, most DNA is read, is expressed, and yields protein. And since prokaryotes have a very large panel of energy-dependent proteases which patrol the cell and degrade almost anything that's not properly structured, we can also assume that if something is expressed in a prokaryote, it's almost certainly protected from these proteases, and the mechanism we know which yields protection is structure. Right, and so seeing these things and thinking about these things, I started to look at repetition. Where does repetition occur in prokaryotic genes where I'm reasonably confident that these genes are protein coding? And here is an example. It's a pretty obvious example. In fact, the structure you see on the right side is the crystal structure of the sequence. You might be struck, it's a beta propeller, of course. You might be struck by the fact that the individual blades of the propeller are virtually identical, right? There's only a small number of amino acid changes highlighted in yellow. Everything else is the same between all the blades. And if you go to the DNA level, you see that there are no synonymous changes. All the changes are non-synonymous and yielded these amino acid changes. So you can't even do a DN over DS analysis because there's no synonymous mutations. This thing was clearly amplified very recently. In fact, looking at the origin of the different strains, this was probably amplified within the past 100 years. So clearly, life generates new folded proteins because the last relationship of this amplified propeller to other proteins in our proteome is at the level of a single blade. But as we have published a few years ago, propellers that are more than 90% sequence identical can generally be seen to have been amplified independently of each other. And that's because their DNA sequence shows small idiosyncrasies for each of the propellers, showing that they were amplified from a single blade. And so, yes, I take this as evidence that nature keeps producing new folded proteins, but there are proteins which follow a prototype that is as old as the last common ancestor. Beta propellers originated probably close to 3.6, 3.7 billion years ago. And so nature is making new exemplars of these repetitive protein domains, but they are not new domains. They are still the same ancient fold that we have seen in life across the hundreds of millions of years. All right, we can see the same thing, for example, in these Hendekad coiled coils. Now, typically you might know that coiled coils are generally repeats of 7 residues, the so-called heptad repeat. But here we have a family of fungal proteins, which are actually very widespread in fungi. And the exemplar on the left from Ustillagoi, Ustillaginoidea virens. It has a Hendekad coiled coil. You see here the repeat. And the one from Metarhizium has a heptad coiled coil. So the flanking regions are homologous, not identical. Here in gray, I have highlighted the differences between this protein and this protein. And you see the distance is substantial, but they are homologous. But the stalk, the coiled coil, is not homologous. It was amplified separately in this protein from this protein. And you see that the heptads here, by and large, are identical, and the Hendekads here, by and large, are identical. Now, if we use AlphaFold, you can see that the proteins are substantially the same. Except the supercoil is different because Hendekads are much straighter than heptads. But other than that, it's the same protein, but because the middle was amplified separately, it's not quite the same protein. It's homologous in parts and analogous in other parts. And so here again, we see that nature has created variants of existing proteins, but they are not new proteins in the sense of presenting us with new folds and new activities. They're still very much the same proteins that had been there many, many millions of years ago. All right. Oh, why is this here? Okay, I'm not sure why that slide is in there. It doesn't belong there. Now, the point I want to touch now is the fact that these repetitions are very widespread also in prokaryotic genomes, and some of them are clearly expressed open reading frames, like this protein from Ustillaginoidea virens. And here in gray, I have marked the differences between the individual repeats, showing that they have evolved for a certain distance. And also here at the amino acid level, showing that the protein has undergone evolution. It has many homologs. We have seen them. So this is clearly a protein, and it's just the middle part that has been amplified. But if we look at something like this, we see that at the protein level, the repeats, there are also Hendekads, and the repeats are identical. At the DNA level, we see that there's a small number of differences, but they are silent, and at the amino acid level, you don't see them. In any case, here as well, AlphaFold predicts a coiled coil. In fact, with very good PLDDT values. That's why we included it in our paper. But I'm not absolutely sure that this is a real protein, an ORF that encodes a protein. I assume it is because there's a few synonymous mutations, but we haven't experimentally tested whether there's a promoter, of course. But here we have a protein where or an ORF where the repeats are identical at the DNA level and the protein level. And here it's absolutely possible that this is a virtual protein, that is an open reading frame that could become a protein if it had a promoter, but quite possibly it is not a protein coding gene at this time. Again, AlphaFold is very confident in predicting an antiparallel for helical coiled coil with PLDDT values between 70 and 90. But we see in these three examples a sort of continuum which might track the birth of new protein coding genes in these organisms. And so based on these observations, I started to wonder whether we can expect new folded proteins from such ORFs based on the power of repetition to actually generate structure. And so basically here I've recapitulated a few points. Yes, most proteins in our proteome go back to exemplars that originated before the last common ancestor. However, almost every genome contains many open reading frames which may encode proteins but don't have recognizable homologs in other genomes. And some of these orphans, possibly many, have repetitive sequences. And since repetition is so powerful in generating protein structure, I started to wonder whether we might see new proteins arising from this dark genomic matter. And if yes, what structures might these new proteins form? And of course, since I was looking for repeats that are fairly short, like heptads and hendecads or tetrads, I would assume that they're all fibers, right? And so one starting point for my searches was this protein shown right here. It's a synthetic nanotube of coiled coil sheets in which the helices have essentially 180-degree angle between the interfaces on the two sides. So if you look at this helix here, it has an interface to this helix, and it has an interface to this helix. And the angle between these two interfaces, as shown here, is about 180 degrees. And this is a synthetic peptide generated in order to obtain such alpha sheets, because the most open arrays of alpha helices, which we know that are built on heptads, are decameric. And so in a decamer, you span about 144 degrees here, whereas in this open-ended sheet, which can go on for many nanometers, you have practically 180 degrees. And so I was wondering, since the sequence of this designed protein was fairly simple, whether nature was also doing this. And so here you see the sequence, which was designed in order to form nanotubes, helical nanotubes. And the key to this sequence is the fact that it is built of heptads, and the heptads have alanine in the so-called position A of the heptad and leucine in position D. And then they have isoleucine in position C and alanine in position F. Now, let's look at the helical wheel, what that means. And what we see is that we have alanine. No, where's here? Yeah, we have alanine, then connecting to, excuse me, we have alanine connecting twice leucine to leucine, and then again to alanine. Alanine connecting twice leucine to leucine, and again to alanine. And this arrangement places the alanines at opposite ends of the helical wheel, and the leucine and the isoleucine flanking them and able to form an interface with them. However, an interface where these residues being substantially bulkier than these residues, that could be 180 degrees. And in fact, here you can see by the fact that the alanines have considerably smaller bulk than isoleucine and leucine, you can achieve a side-by-side packing of these helices with an angle between these two interfaces of about 180 degrees. And this is not speculation because you've seen the structure, and that's in fact what these form. So I was wondering, this is so incredibly simple. Are there natural proteins which do this? And so I found this protein, navigating the proteome of life. I ran into this and I immediately saw the pattern in the C-terminal part, right? And you can see it, alanine, valine instead of isoleucine, leucine, and alanine. And so this, in principle, would look a lot like the alpha sheet made by Vince Conticello at the University of Georgia. And here you see a comparison of this sequence against the structure that Vince made. And you see it's 50% sequence identical, and all the key residues are in the correct places. So what does AlphaFold say about this? Well, it says, yeah, it could be more or less a sheet. Now, AlphaFold doesn't necessarily do very well with fibrous proteins, but in this case, the helices are not that long and they don't look all that fibrous. So, and the PLDDTs were in the upper 80s. So I tended to agree with this prediction. We tried to make the protein, we didn't succeed. It doesn't express in E. coli. And so we would need to screen through expression systems to find one that will do it. It hasn't been a priority so far. But in seeing this, I realized that a few other proteins that I had looked at, which also had two ledges, but the alanines were not convincing. It's mostly here, but the others are not really alanines. But the structure looks surprisingly similar, making me wonder whether I'm looking at a more general tendency of highly repetitive alpha helices to array in various supramolecular architectures. Now, here is, so this was my first example, alpha sheets. Here is another one that I've seen several of. And this is recently amplified protein. If it is a protein, the entry was retired from NCBI. So during reannotation, this was deleted. But I see this pattern of tryptophan here, then X leucine, X alanine, in lots of other things. And then, of course, ending on glycine. And so I looked around, and here you can see these repeat units from other hypothetical proteins. I don't think there's anything homologous here. I think that these are just some structural solutions that life has kept running into. And the key for me was the tryptophan here and the glycine at the end. And having a zigzag of hydrophobics, with the first hydrophobic larger than the second hydrophobic, suggested to me that this might be a beta helix of the, let me think, of the right-handed type. And indeed, AlphaFold tells me that it's a beta helix of the right-handed type with the tryptophans and the leucines in the core and 3 sides to the beta helix. And then the end here, which looks like a Hendekad coiled coil, actually is predicted to be a Hendekad coiled coil. And so could it be that the constant popping up of this motif is due to it being a particularly good motif for this kind of beta helix? I don't know, maybe. But looking at the gene invariant of these ORFs, I'm not even convinced that all of them are being expressed. So this looks like a virtual protein which sometimes can acquire a promoter and become expressed. Right, my last example, and then I'll come to a conclusion. My last example is a protein which I became interested in because it seemed to be similar, but in an analogous way to the blades of a beta propeller. I found that super interesting. Now, this protein has also been retired from NCBI, but not from UniProt. So I have no idea whether it's even still thought to be a protein. But if you look at the repeats, which I've listed here, they're not completely identical. So to me, that suggests that this is a protein and it has already undergone some evolution. But maybe I'm wrong. In any case, what does AlphaFold do with this? We tried to express it, but we failed. E. coli doesn't want to express this. And we haven't gone through expression systems. But what does AlphaFold do with it? Well, definitely not beta propellers. It makes this sort of amyloid-like strange beta helix. Now, I was intrigued when I saw it, but I'm not nearly as intrigued now after many colleagues have observed that if you give AlphaFold a highly repetitive sequence, it will tend to put it into some sort of a beta helix arrangement. Certainly, if it can't plausibly make an alpha helix, it'll make various forms of beta helices. So I think this is a real protein, and if it is a real protein, its structure will be highly repetitive. But right now, I don't have any concrete evidence. All right, so what does this tell us? Where does it leave us? First, I should say that if I can think of something, nature has tried it. So if it can be done, I can probably find exemplars by looking through natural protein sequences. And that's maybe one of the reasons why I spend so much time roaming around in sequence space and just looking at things and trying to wrap my mind around what the genomes of organisms are encoding. And here I look primarily at prokaryotes because, as I said, most of their open reading frames are coding And most of the proteins that are expressed are folded, right? And so I have many more odd exemplars that I could share with you, but I think that I've made my point. I would emphasize that nature keeps teaching us, but we very often don't listen, which means that sometimes it has been telling us the same thing for years, and we just did not follow what nature was telling us. And in principle, if we learn how to listen more carefully and patiently, we might start seeing a lot of things that we currently don't place because they're outside our experience. Let me just mention the CRISPR-Cas system, which was found initially by Japanese researchers through the repeating segments in prokaryotic DNA, right? For quite a while, people didn't know what to make of it. For pretty good reason, the logic of those systems was outside our experience. So if we look at nature in an unbiased way, we might see a lot of things that are outside our direct experience. Now, more concretely to the topic of today, I think that new repetitive folded proteins arise all the time. Some of them replicate folds that were already present for extraordinarily long periods of time, and those may become established for a certain time before they die out again. But most of these new folded proteins will disappear almost as soon as they appeared, because they will mainly yield biomaterials, but not show any chemical activity, unless the cell needs some sort of a new collagen or a new cytoskeleton, which typically it won't. These proteins will disappear as soon as they have appeared, right? And so if we look at the proteome of life today, we might see things surfacing briefly in a few or even in a single strain and then disappearing again. And this, I've come to be convinced, is as much an evolutionary reality as these extraordinarily permanent proteins, structured proteins that form the essence of our proteome, right? So the lack of new proteins is not due to our failure to generate them in living systems. It's due to the fact that they can't find an ecological niche and die out almost as soon as they arose. All right, and this brings me to the end of what I wanted to tell you, and I'm very happy if you have questions for me.